In 1994, a physicist named Edward Witten stood before a group of his peers at the University of Southern California. He was there to talk about string theory. At the time, there were 5 competing versions of it. It was a mess, but Witten proposed something new. He suggested a single unifying theory. He called it M theory. The M could stand for mystery or magic or mother, he said. An M theory had a strange requirement. It needed 11 space-time dimensions to work. We only see 4. 3 of space and 1 of time. Sir Witten explained that the other 7 dimensions are there. They are just curled up, incredibly small. Too small for us to ever perceive directly. His idea changed the field of physics. It showed that the reality we experience might just be a tiny sliver of a much larger, stranger universe. This way of thinking, that a fundamental truth is hidden in plain sight, applies to another big question. The question of where we come from. The standard story is that life began on Earth. In some warm little pond or a deep sea vent, a chance combination of chemicals zapped by lightning or heated by magma sparked into the first living thing. This is called abiogenesis. It's a solid, respectable theory, but it has problems. The big ones, the jump from non-living chemicals to a living, replicating cell is enormous. We haven't been able to repeat it in a lab. The timeline is also tight. Earth formed about 4.5 billion years ago. For a while, it was a molten hellscape. The oldest evidence of life we have is from around 3.8 billion years ago. That does not leave much time for such a complex event to happen by chance, so some people look elsewhere. They look up. They wonder if the seed of life wasn't homegrown. They wonder if it came from somewhere else. This idea is called panspermia. It suggests life is common in the universe. It travels between stars and planets, riding on comets or asteroids. Earth was not a sterile cradle. It was a fertile garden just waiting for a seed to land. This idea is not new. It has been around for over a century. But for a long time, it was on the fringe. It was seen as science fiction. Now, things are changing. We are finding more and more evidence that makes panspermia seem less and less crazy. We find organic molecules, the building blocks of life, in meteorites. We find water on Mars and moons of Jupiter. We discover tiny organisms on Earth that can survive the vacuum of space. The idea that life began somewhere else and arrived here is moving from the fringe toward the mainstream. It forces us to reconsider our place in the cosmos. Maybe we are not a unique accident. Maybe we are part of a vast, interconnected web of life that spans the galaxy, just like those extra dimensions Witten talked about. Our true origins might be hidden from our immediate view, locked away in the vastness of space. Number 15, life came here on a rock. On any given day, over 100 tons of material from space slams into Earth's atmosphere. Most of it is dust, tiny specks that burn up harmlessly, but some of it is bigger. Rocks that were blasted off other planets millions of years ago. A large enough impact on Mars could send debris flying into space. Some of that debris could, after a long journey, find its way to Earth. It has happened. We have found over 277 meteorites on Earth that we know for certain came from Mars. We know this because they have gas trapped inside them. The composition of that gas perfectly matches the Martian atmosphere, which we measured with the Viking landers in 1976. The journey can take a long time. The famous Mars meteorite ALH-41 traveled for about 17 million years before it landed in Antarctica. This is the core idea of lithopanspermia. Life travels inside rocks. Litho means stone. The rock acts as a spaceship. It shields its tiny passengers from the worst of space. The vacuum is not a problem for many microbes. The cold is also manageable. But the radiation is a killer. Space is flooded with cosmic rays and solar radiation that shred DNA. A few feet of rock, however, can block a lot of that radiation. The journey itself is possible. The ejection is the first hurdle. An asteroid impact on a planet like Mars would be catastrophic. But not everything at the impact site gets vaporized. The edges of the crater get thrown outwards at high speed. It is a process called spallation. It can launch rocks into space without heating them up enough to sterilize them. And experiments have shown that microbes can survive the immense shock forces of such an event, pressures up to 50 gigapascals. That is hundreds of thousands of times Earth's atmospheric pressure. The second hurdle is the long journey through space. Millions of years in the freezing, irradiated dark. It sounds impossible, but some bacteria on Earth can go dormant. They form spores, they shut down all their biological processes, they can wait like that for an incredibly long time. In 2000, scientists claimed to have revived bacteria from a 250 million year old salt crystal found in New Mexico. While that claim is debated, it points to the potential for long-term survival. The radiation dose over millions of years would still be a problem. But if a microbe was deep inside a large rock, its chances would improve. The final hurdle is landing. A rock hitting Earth's atmosphere at 25,000 miles per hour gets hot. The outside melts and burns away. This is called ablation, but it happens very fast. The heat does not have time to penetrate deep into the rock. The inside can remain cold. Think of a steak seared on a grill. The outside is cooked, but the inside is still rare. If a meteorite is big enough, say the size of a fist or larger, its core can stay at survivable temperatures. It then hits the ground, breaks open, and releases its cargo. If it lands in a suitable environment like an ocean or a lake, the microbes could wake up and start to multiply. This is not just a theory. Scientists have tested it. The Biopan facility is a European Space Agency experiment. They attach it to the outside of satellites. They put bacteria and other microorganisms inside, sometimes mixed with clay or rock dust. They expose them to space for a year or two, then bring them back. Many of the organisms survive. The tardigrade, a tiny creature also known as a water bear, is famous for its resilience. In 2007, they sent some tardigrades into space on a Biopan mission. They were exposed to the pure vacuum and intense radiation of space with no protection. When they returned to Earth, many of them were revived. They even, went on to reproduce. If a microscopic animal can survive raw space, a bacterium tucked inside a rock has a much better chance. The idea that life hitched a ride on a piece of Martian rock is not so far-fetched. This early Mars was likely warm and wet. It may have been a better place for life to start than early Earth. So maybe, just maybe, we are all Martians. Number 14, life arrived in a dirty snowball. Comets are not like asteroids. Asteroids are mostly rock and metal. Comets are a mix of ice, dust, and rock. People call them dirty snowballs. They come from the outer edges of the solar system, from places like the Kuiper Belt and the Oort cloud. They are ancient. They are leftovers from the formation of the solar system 4.6 billion years ago. They are like time capsules. They contain the original materials that built the planets. For a long time, we thought comets were just frozen chunks of water and rock. But we were wrong. In 2014, the European Space Agency's Rosetta mission reached Comet 67P Churyumov. It was an incredible achievement. The spacecraft orbited the comet for 2 years. It even sent a small lander fly down to the surface. What Rosetta found changed our understanding of comets. It found complex organic molecules. These are the carbon-based molecules that are essential for life. It found glycine, the simplest amino acid. Amino acids are the building blocks of proteins. It found phosphorus, a key component of DNA and cell membranes. It even found the organic molecule acetone, which is used as a solvent. The comet was not just a dirty snowball, it was a flying chemical factory. This supports a version of panspermia called cometary panspermia. The idea is that comets, not asteroids, are the primary delivery system for life, or at least its ingredients. Comets have a big advantage over rocks. They contain huge amounts of water ice. When a comet approaches the sun, the ice turns to gas, creating the beautiful tail we see. Early Earth was very hot and dry. The theory of the late heavy bombardment suggests that for a period of about 400 million years, ending around 3.8 billion years ago, Earth was pelted by a huge number of asteroids and comets. This bombardment could have delivered the water that now fills our oceans. If it could deliver water, it could deliver other things too. A comet's interior is a promising place for life's chemistry to happen. It is shielded from radiation. It has water ice. It has organic molecules. It has clay particles, which can act as catalysts, helping to assemble more complex molecules. The interior of a comet is not completely frozen. The decay of radioactive elements like aluminum-26 could have created pockets of liquid water inside comets for millions of years. These pockets would have been like tiny protected laboratories. All the ingredients for life would be there, sloshing around in the dark. It is possible that life itself, not just its building blocks, could form inside a comet. When a comet hits a planet, the impact is immense, but not all of it would be sterilized. Some comets might break up in the atmosphere. They could release vast clouds of dust and water vapor. This dust, containing organic molecules or even dormant microbes, would then gently settle all over the planet. This is a much softer delivery method than a solid rock slamming into the ground. The famous astronomer Fred Hoyle was a huge proponent of cometary panspermia. He and his colleague Chandra Wickramasinghe argued that life did not just arrive on Earth once. They believed it is still arriving. They suggested that new viruses and bacteria are constantly raining down on us from cometary dust. They even blamed this cosmic influx for disease outbreaks like influenza pandemics. Most scientists reject these specific claims, but the core idea that comets are rich in the stuff of life is now widely accepted thanks to missions like Rosetta. Whether they delivered just the ingredients or the finished product is still an open question. But the dirty snowballs of our solar system look a lot more like life-giving oases than we ever thought. Number 13, a meteorite in Australia held the secret. On September 28, 1969, a fireball lit up the sky over the small town of Murchison, Australia. A large meteorite had entered the atmosphere and broken apart. The pieces rained down over a 5-square-mile area. People collected the fragments. They were dark, and they had a strange tar-like smell. The Murchison meteorite, as it came to be called, turned out to be one of the most important rocks in scientific history. It was a carbonaceous chondrite, a type of stony meteorite that is rich in carbon and has not been significantly altered by heat. It was a pristine sample of the early solar system. Scientists at NASA and other institutions began to analyze the Murchison meteorite, and what they found was stunning. The rock was packed with organic compounds. They identified over 100 different amino acids. Our bodies use just 20 amino acids to build all our proteins. The meteorite contained many of those, like glycine and alanine, but it also contained dozens of others that are not found in life on Earth. This was a crucial clue. It proved the amino acids were not from contamination after the meteorite landed. They were truly extraterrestrial. Another important clue was the handedness of the molecules. Many organic molecules, including amino acids, can exist in two forms. They are mirror images of each other, like your left and right hands. They are called enantiomers. Life on Earth exclusively uses left-handed amino acids. Why this is, nobody knows. It is one of the great mysteries of biology. When scientists synthesized amino acids in the lab, they always got a 50-50 mix of left-handed and right-handed versions. The Murchison meteorite also had a mix. But there was a slight excess, about 9% of the left-handed kind for some of the amino acids. This was a bombshell. It suggested that something in space favors the formation of left-handed amino acids. One theory is that polarized light from neutron stars or supernovae could selectively destroy right-handed molecules in the interstellar dust clouds where meteorites form. If meteorites like Murchison were constantly falling on the early Earth, they would have seeded the planet with these left-handed dominant amino acids. This could have given life the starting bias it needed. It would solve the mystery of why all life on Earth shares the same molecular handedness. The meteorite did not just contain amino acids, it also contained other building blocks of life. It had purines and pyrimidines, which are components of DNA and RNA. It had fatty acids, which can form cell-like membranes called micelles when in water. In 2020, an even more incredible discovery was made in the Murchison meteorite. A team of scientists found sugars, including ribose. Ribose is the R in RNA. It is the backbone of the molecule that many scientists believe was the precursor to DNA. Finding a key component of RNA in a 4.6 billion-year-old rock from space is a profound discovery. The Murchison meteorite does not prove that life came from space, but it proves that the building blocks of life are abundant in space. It shows that the raw materials for creating a living cell were delivered to the early Earth for free. So it was not necessary for Earth to create all these complex molecules from scratch in some primordial soup. The soup was being seasoned from above. This idea is sometimes called pseudo-panspermia or molecular panspermia. It is the theory that the components of life, rather than life itself, came from space. It is the most conservative and widely accepted version of the panspermia family of theories. The smelly black rock that fell on Australia in 1969 provides the strongest physical evidence we have that our planet's biology has deep cosmic roots. Number 12, a Martian rock in Antarctica sparked a global debate. In December 1984, a team of American meteorite hunters was working in the Allen Hills region of Antarctica. The vast white ice sheet is a perfect place to spot dark rocks from space. A scientist named Roberta Score picked up a greenish-gray rock that weighed about 4 pounds. It was logged as ALH 8401 for years. It sat in storage, misidentified as just another common type of meteorite. But in 1993, scientists re-examined it. They realized its chemical composition was unique. It was a Martian meteorite, and it was incredibly old. It formed on Mars about 4.1 billion years ago, a time when Mars was thought to be warmer and wetter. It was blasted off the planet by an impact 17 million years ago and landed on Earth about 13,000 years ago. A team at NASA's Johnson Space Center, led by David McKay, began to study the rock in detail. In 1996, they published a paper in the journal Science that made headlines around the world. Well, they claimed to have found evidence of fossilized microbial life inside the Martian rock. The President of the United States, Bill Clinton, even gave a speech about the potential discovery. It was a moment of global excitement. Life on Mars seemed within our grasp. McKay's team presented 4 main lines of evidence. First, they found complex organic molecules called polycyclic aromatic hydrocarbons, or PAHs. These can be produced by the decay of living organisms. Second, they found minerals like iron sulfide and magnetite in close proximity. On Earth, certain bacteria produce these minerals. Third, they found tiny carbonate globules inside the rock. These structures could have been formed by biological activity in water. The fourth and most compelling piece of evidence was a series of electron microscope images. The images showed tiny, segmented, worm-like shapes. They looked remarkably like fossilized bacteria, only much smaller than any known bacteria on Earth. The announcement was met with intense scrutiny and skepticism from the scientific community. Other scientists began to pick apart the evidence, one piece at a time. They showed that PAHs can be formed by non-biological processes. They are found in all sorts of meteorites and even in interstellar dust. The minerals could also have been created by high temperatures and pressures, perhaps from the impact that launched the rock into space rather than by Martian microbes. The carbonate globules, too, could have a geological, not biological, origin. The tiny nanofossils were the most controversial part. Other scientists argued they were far too small to have ever been living cells. They were smaller than the theoretical minimum size for a cell to contain all the necessary machinery like DNA and ribosomes. And critics suggested they were just unusually shaped mineral crystals or artifacts of the imaging process. The debate raged for years. For every paper published supporting the biological origin, another would be published refuting it. Today, the consensus is that ALH 84001 does not contain conclusive evidence of Martian life. Most scientists believe that all the features observed in the rock can be explained by non-biological processes. But the debate was not a failure. It was a huge success for science. It forced scientists to think hard about what constitutes evidence for life. It led to the development of new techniques for analyzing samples and avoiding contamination. It spurred a new era of Mars exploration with missions like the Spirit, Opportunity, and Curiosity rovers, specifically designed to search for signs of past or present life. The story of ALH 84001 is a cautionary tale about jumping to conclusions, but it also cracked open the door to the possibility that we might one day find a fossil in a rock from another world. It made the idea of life beyond Earth feel real and tangible, something we could actually hold in our hands. Number 11. The toughest creatures on Earth are spacemen. There is an animal on Earth that can survive being boiled, frozen, crushed, and starved. It can withstand radiation doses 1,000 times greater than what would kill a human. It can survive the vacuum of space. It is called the tardigrade, or water bear. It is a microscopic, 8-legged invertebrate that lives in moss and lichens all over the world. Its abilities are so extreme, they seem to come from science fiction, but they are very real, and they provide some of the best evidence that life could survive an interplanetary journey. Their tardigrades survive these extreme conditions by entering a state of suspended animation called cryptobiosis. They curl up into a little ball called a ton. They retract their legs, lose over 97% of their body's water, and shut down their metabolism to almost nothing. In this state, they can persist for decades. When they are rehydrated, they can wake up and go about their business as if nothing happened. The key to their survival is a special type of protein in their cells. These proteins form a glass-like substance that protects their cellular structures from damage when they dry out. It is a process called vitrification. In 2007, the Photon M3 mission carried a payload of tardigrades into low Earth orbit. Some were exposed only to the vacuum of space. Others were exposed to both the vacuum and the full blast of solar UV radiation. After 10 days, the satellite returned to Earth. The tardigrades that were shielded from the UV radiation survived at a high rate. They were rehydrated and went on to lay eggs that hatched normally. Even some of the ones exposed to the deadly UV radiation survived. This was the first time an animal had been shown to survive the combination of vacuum and radiation in open space. Tardigrades are not the only extremophiles, or lovers of extremes, that push the boundaries of life. The bacterium Deinococcus radiodurans is listed in the Guinness Book of World Records as the world's toughest bacterium. It can withstand radiation doses 3,000 times greater than what would kill a human. It has incredible DNA repair mechanisms. If its chromosomes are shattered by radiation, it can stitch them back together perfectly within a few hours. In an experiment on the International Space Station, pellets of this bacterium were exposed to space for 3 years. The ones on the surface were killed by UV radiation, but the bacteria in the lower layers of the pellets, shielded by their dead comrades, survived. The scientists calculated that a pellet of these bacteria just 1 millimeter thick could survive in space for up to 8 years. Inside a rock, it could survive for much longer. There are other extremophiles too. Thermophiles thrive in boiling hot springs and deep-sea hydrothermal vents at temperatures above 100 degrees Celsius. Psychrophiles live in ice and frozen soil in Antarctica at temperatures below freezing, and halophiles live in water that is 10 times saltier than the ocean. The existence of these organisms on Earth dramatically expands the range of conditions where life can exist. It makes it more plausible that life could survive on a frozen world like Jupiter's moon Europa or in the subsurface of Mars. These creatures show us that the barriers to panspermia are not insurmountable. The journey through space is harsh, but not necessarily lethal. Life, once it gets started, is incredibly tenacious. Extremophiles are living proof of concept. They demonstrate that the biological hardware required to survive an interplanetary trip already exists. This does not mean that panspermia happened, but it makes it much harder to dismiss. It is no longer a question of if life can survive in space. The question now is whether it has ever had the opportunity to make the trip. Number 10, mysterious red rain fell over India. In the summer of 2001, a strange phenomenon occurred in the southern Indian state of Kerala. From July 25th to September 23rd, periods of heavy rain were colored a startling red. The rain was localized, often falling over areas of just a few square kilometers. It was so thick it stained clothes pink. At first, people thought it was pollution from a local factory, but the rain was widespread. The local government initially suggested the color was caused by dust from a meteor that had exploded in the atmosphere. This explanation was quickly dismissed by mainstream scientists. They concluded the color came from airborne spores of a local type of green algae, Trentipolia annulata. Case closed. But Godfrey Lewis, a physicist at Cochin University, was not convinced. He collected samples of the red rain. He examined the particles under a microscope. They were not algae spores, he claimed. They were strange red-walled cells. They had no nucleus, no DNA. They were about 4 to 10 micrometers in size, about the size of a red blood cell. Whereas they looked like biological cells, but they were unlike any known cells on Earth. Lewis and his student, Santosh Kumar, performed a series of experiments. They found that the cells were inert at room temperature. But when they heated the cells to 121 degrees Celsius, smaller daughter cells appeared to emerge from the parent cells. The cells seem to be reproducing, but only at very high temperatures. This is characteristic of extremophiles. Lewis put forward a radical hypothesis. He suggested the red cells were extraterrestrial in origin. He revived the meteor explosion theory. He proposed that a comet or meteorite carrying these microbial cells had disintegrated over Kerala, seeding the clouds with its cargo. The cells then came down in the rain. His theory was published in the fringe journal Astrophysics and Space Science in 2006. It was mostly ignored by the mainstream scientific community. The algae spore explanation was considered sufficient. However, the story did not end there. Other researchers took an interest. In 2013, a team of scientists, including Chandra Wickramasinghe, the famous proponent of panspermia, analyzed the cells again. They confirmed the absence of DNA. This is extremely unusual. All life on Earth uses DNA to store its genetic information. Finding a cell without it would be a monumental discovery. They claim to have detected DNA using a fluorescent dye called DP, but the results were ambiguous and have been disputed. In 2015, another group of researchers published a paper claiming to have finally sequenced the DNA from the red rain cells. Their results showed it was indeed the DNA of the algae Trentipolia annulata. So, was it just algae? The case is still not completely settled. The proponents of the extraterrestrial hypothesis argue that the DNA found was contamination. They point to the unusual properties of the cells, like their ability to reproduce at high temperatures and their lack of DNA as evidence that they are something new. The vast majority of scientists believe the algae explanation is correct. It is the simpler and more logical conclusion. But the red rain of Kerala remains a fascinating anomaly. It is a powerful example of how an unexplained event can fuel speculation about panspermia. Even if the cells do turn out to be a common earthly algae, the incident highlights how easily microscopic life can be swept up into the atmosphere and distributed over wide areas. It shows a plausible mechanism for how extraterrestrial cells, if they were to, enter our atmosphere, could be delivered to the surface. The mystery of the red rain is a reminder that strange things still fall from the sky. Number 9. Life is deliberately sent by an alien civilization. In 1973, two brilliant and famous scientists proposed a version of panspermia that sounded like pure science fiction. The scientists were Francis Crick, the co-discoverer of the structure of DNA, and Leslie Orgel, a leading researcher on the origin of life. They were both giants in their fields. They published a paper titled Directed Panspermia. They argued that life on Earth might not have arrived by accident. It might have been sent here on purpose by an advanced alien civilization. Crick and Orgel were motivated by two main problems with the standard origin of life theories. First, the timeline seemed too short for life to arise by chance on Earth. Second, they were puzzled by the universal genetic code. All life on Earth, from bacteria to humans, uses the same DNA code to translate genes into proteins. This suggests all life shares a single common ancestor. But they were also struck by the dependence of life on certain elements, like molybdenum, which is relatively rare in the Earth's crust. They argued it was strange for life to evolve to depend on a rare element, so they proposed a wild alternative. Billions of years ago, a highly advanced civilization elsewhere in the galaxy faced a crisis. Perhaps their star was about to die, or their planet was becoming uninhabitable. They could not travel to other stars themselves, but they wanted their legacy to survive. So they built a spaceship, they loaded it with a payload of microorganisms, perhaps a diverse sample of bacteria. They chose hardy microbes that could survive a long journey in a dormant state. They aimed their spaceship at a distant young star system that had a promising-looking planet, a planet like the early Earth. The spaceship would have been designed to protect the microbes from radiation and the cold of space. After a journey of millions of years, it would arrive at its destination. It would then release its cargo into the planet's atmosphere or ocean. These microbes would then begin to grow and evolve, eventually leading to all the life we see today. We would be the descendants of those alien microbes. Crick and Orgel were careful to point out that this was just speculation. They had no direct evidence, but they argued it was a plausible scientific hypothesis that should be considered. It solved some problems. It explained how life could appear so quickly on Earth. The hard work of creating life from scratch was done somewhere else. It also could explain the molybdenum issue. Maybe the planet where life originated was rich in molybdenum. The aliens would have sent microbes that were already adapted to using it. The theory of directed panspermia is, of course, impossible to prove or disprove right now. It raises many more questions than it answers. Who were these aliens? Where did they come from? Why did they do it? Were they benevolent gardeners seeding the galaxy with life? Or was it an accident, a form of cosmic pollution from their garbage? The theory also just pushes the problem of the origin of life back one step. It explains how life got to Earth, but not how life started in the first place. Despite these issues, the idea has a certain appeal. It is the ultimate message in a bottle story. The message is life itself. Some have even suggested that the aliens might have encoded a message within the DNA of the microbes they sent. The idea of a sentinel gene or a sequence that could only be artificial is intriguing. Scientists have searched for such a message in our own DNA but have found nothing. Directed panspermia remains on the far fringes of science, but the fact that it was proposed by scientists of the caliber of Crick and Orgel means it cannot be dismissed entirely. It is a thought-provoking idea that forces us to confront the possibility that our existence may not be a random accident, but the result of a deliberate act by some long-dead cosmic intelligence. Number 8. Cosmic rays can push life between stars. One of the oldest versions of the panspermia theory does not involve rocks or comets. It involves something much smaller, individual microbes or spores floating freely in space. The idea was first proposed in detail by the Swedish chemist and Nobel laureate Svante Arrhenius in 1903. He called it radiopanspermia. He suggested that tiny particles like bacterial spores could be lifted from the upper atmosphere of a planet with life. Once in space, they could be pushed by the pressure of starlight. Light exerts a tiny physical pressure. It is called radiation pressure. For large objects, this pressure is negligible. But for a microscopic particle, it can be a significant force. Arrhenius calculated that if a particle was small enough, between 0.2 and 1.5 micrometers, the pressure from the sun's light would be strong enough to overcome its gravity. The particle would be pushed away from the sun and out into interstellar space. It would travel like a tiny solar sail. This tiny spore could then travel for millions of years. Eventually, it might drift into another solar system. The gravity of a star or a planet could capture it. It could then fall onto a habitable planet and begin to grow. In this way, Arrhenius imagined life could spread from star to star, like dandelions spreading their seeds on the wind. It was a beautiful and elegant idea. However, modern science has identified a major flaw in the theory of radiopanspermia. Space is not empty. It is filled with deadly radiation. The biggest problem is ultraviolet UV radiation from stars. A naked bacterial spore, unprotected by rock, would be exposed to the full force of this radiation. UV light is very effective at sterilizing things because it damages DNA. A spore floating in space would be sterilized in a matter of hours or days, not the thousands or millions of years needed for interstellar travel. There is also the problem of cosmic rays, which are high-energy particles that would also damage the spore's DNA. Because of this radiation problem, most scientists abandoned the idea of radiopanspermia, where life is shielded inside a rock, seemed much more cool, but the idea is not completely dead. Some researchers have suggested ways to overcome the radiation problem. Perhaps the spores are coated in a protective layer of carbon, like graphite. This could absorb the UV radiation. Carl Sagan, the famous astronomer, pointed out that this carbon layer would also make the particles black, which would help them absorb the momentum from starlight more efficiently. Another idea is that life travels not as single spores, but in clumps or mats. A clump of bacteria, even a small one, could have a dead layer on the outside that shields the bacteria in the interior. The outer layer gets fried by radiation, but the inner layer survives. If the clump is big enough to offer protection, it might be too big to be pushed effectively by starlight. This is the central dilemma of radiopanspermia. Despite its problems, radiopanspermia has a certain grandeur. So it suggests that the galaxy is filled with a fine mist of life spores constantly drifting between the stars. Any planet that is remotely habitable will eventually be seeded. This would mean that life is not a rare accident, but an inevitable consequence of cosmic physics. It would also mean that all life in the galaxy might share a common ancestry. The life on a planet orbiting Alpha Centauri could be our distant cousin. While the specific mechanism proposed by Arrhenius seems unlikely, the general idea of life being transported on a microscopic scale is still being explored. For example, dust grains coated in ice and organic molecules are common in space. It is possible that these grains, rather than naked spores, could be the vehicles for interstellar panspermia. Number 7. Life hops between planets in the same solar system. The vast distances between stars are a major challenge for any panspermia theory. An interstellar journey would take millions of years, with a high risk of radiation damage. But what if life does not need to cross the interstellar void? What if it just hops between neighboring planets? This is the idea of ballistic panspermia, also known as interplanetary or local panspermia. It is the most conservative and most likely form of panspermia to be true. The theory suggests that life can be transferred between planets within the same solar system. The mechanism is the same as lithopanspermia. An asteroid impact on one planet ejects rocks containing microbes into space. These rocks then orbit the sun until they are captured by the gravity of another planet. The key difference is that the travel times are much shorter. A trip from Mars to Earth can take as little as a few months, although it usually takes millions of years. But even a journey of a few million years is much shorter and safer than a journey of hundreds of millions of years between stars. There is strong evidence that this kind of material transfer happens. We have found meteorites from Mars on Earth. We have also found meteorites from the moon. It is almost certain that rocks from Earth have been blasted into space and have landed on Mars, the moon, and even Venus. A study in 2013 estimated that over the last 3.5 billion years, about 10 billion tons of rock have traveled from Earth to Mars. The exchange of material is a two-way street. This raises an intriguing possibility. Did life start on Earth and then travel to Mars, or did it start on Mars and travel to Earth? There are good reasons to think that Mars might have been a better place for life to begin. About 4 billion years ago, Mars was warmer and had liquid water on its surface. It had a thicker atmosphere. It was a more hospitable environment than the early Earth, which was still being pummeled by giant impacts, including the one that formed the moon. Mars is also smaller than Earth. It has weaker gravity. This makes it much easier to launch rocks into space with an impact. It takes less energy to escape Mars than it does to escape Earth. So a plausible scenario is this life arose on Mars around 4 billion years ago. The planet was a thriving microbial world. Then a large asteroid hit Mars. It sent rocks full of Martian bacteria into space. Some of these rocks traveled to Earth, which was just starting to cool down and become habitable. The Martian microbes found a new home and began to flourish. Meanwhile, back on Mars, the climate changed. The planet lost its atmosphere, got cold and dry, and the life on the surface died out. The life on Earth continued to evolve, eventually leading to us. In this scenario, we are the descendants of Martian life. We are the aliens. This theory is testable. If we find evidence of life on Mars, either fossilized or still living, we can compare it to life on Earth. If Martian life has a completely different biochemistry, with different amino acids or a different genetic code, then it would be a separate second genesis. But if Martian life uses the same DNA and the same left-handed amino acids as we do, it would be strong evidence for a common origin. It would be almost impossible for two independent origins of life to end up with the exact same system. In that case, the principle of Occam's razor, that the simplest explanation is usually the best, would suggest that one planet seeded the other. Given how much easier it is to get rocks from Mars to Earth than the other way around, the Martian origin of life on Earth would become the leading theory. The search for life on Mars is not just a search for aliens. It is a search for our own family tree. Number 6. Viruses are alien invaders that drive evolution. Viruses are strange. They are not really alive. They cannot reproduce on their own. They are just a piece of genetic material, DNA or RNA, wrapped in a protein coat. They need to infect a living cell and hijack its machinery to make more copies of themselves. They are the ultimate parasites. They are also incredibly abundant. There are 10 times more viruses on Earth than there are bacteria. They are found everywhere, from the deep oceans to the upper atmosphere, and for a long time we have thought of them only as agents of disease and death. But what if their role is much bigger? What if they are a major driving force of evolution? And what if they come from space? This is the core of a controversial theory championed by the astronomers Fred Hoyle and Chandra Wickramasinghe. They argued that comets and meteorites are constantly delivering new viruses to Earth's atmosphere. These viruses then fall to the surface and infect living organisms. According to this theory, this constant influx of new genetic information from space is the primary engine of evolution. The standard neo-Darwinian theory of evolution says that new traits arise from random mutations in an organism's DNA, which are then selected by the environment. Hoyle and Wickramasinghe thought this process was too slow and random to account for the complexity of life. They proposed that major evolutionary leaps, like the Cambrian explosion about 540 million years ago when most major animal groups suddenly appeared, were caused by the arrival of new cosmic viruses. These viruses would insert their genes into the genomes of Earth's organisms, creating new body plans and new functions overnight. They even suggested that human evolution was guided by these viral invaders. They pointed to the large amount of junk DNA in our genome, DNA that does not seem to code for anything. They proposed that much of this is the leftover genetic code of ancient viruses that infected our ancestors. Some of this viral DNA, they argued, was later co-opted to create new, uniquely human traits. The theory also attempts to explain diseases. Hoyle and Wickramasinghe famously claimed that flu pandemics are caused by new influenza viruses arriving from space. They pointed out that flu outbreaks often seem to appear simultaneously in widely separated places, which is hard to explain with person-to-person transmission alone. They suggested the virus falls from the atmosphere over a wide area, infecting many people at once. This idea has been almost universally rejected by virologists and epidemiologists. We can track the spread of flu strains around the globe, and they evolve from existing terrestrial strains. They do not appear out of thin air. However, the core idea that viruses play a huge role in evolution is now mainstream. We now know that viral infections are a major source of genetic novelty. A type of virus called a retrovirus can permanently insert its genetic code into the DNA of its host. If this happens in a sperm or egg cell, the viral DNA can be passed down through generations. It is estimated that about 8% of the human genome is made up of these endogenous retroviruses. For a long time, we thought this was just junk. But we are now discovering that some of this ancient viral DNA has been repurposed by our bodies to perform important functions. For example, a protein called syncytin, which is essential for the formation of the placenta in mammals, is derived from an ancient viral gene. Without a virus that infected our distant ancestors, human pregnancy as we know it might not be possible. So, while the idea that new diseases are constantly raining down from space is not supported by evidence, the bigger picture is more nuanced. Viruses are a powerful force of evolution. They do move genes around and create novelty. And the building blocks of viruses like RNA and proteins could certainly exist in space. It is not impossible that a functional virus could be delivered by a meteorite. Whether this has ever happened and had a major impact on the history of life is unknown. But the idea forces us to look at these tiny agents of disease in a new light, as potential cosmic messengers. Number 5. There is a second alien biosphere here on Earth. We search for alien life on other planets. We send rovers to Mars and telescopes to probe the atmospheres of exoplanets. But what if alien life is right here, under our noses, and we just have not noticed it? This is the fascinating and unsettling idea of a shadow biosphere. The theory suggests that life on Earth may have started more than once. The life we know, based on DNA, RNA, and proteins, is just one version. There could be another completely separate form of life sharing the planet with us. It would be a second Genesis. This shadow life would have a different biochemistry. It might use different amino acids, perhaps the right-handed versions that our life ignores. It might use a different set of molecules for its genetic code, something other than DNA or RNA. The physicist Paul Davies, a major proponent of this idea, suggests it might use arsenic instead of phosphorus in its backbone, and its cells would be so different from our own that our standard methods of detection would not work. We look for life by looking for DNA or other familiar biological molecules. If we are not looking for the right thing, we will never find it. This shadow life would be microscopic. It would exist in places where our life does not thrive, in extreme environments that we have not fully explored. Or it could be in the high atmosphere, in deep sea volcanic vents, or locked away in rocks deep underground. It could even be all around us, in the soil and water, but we simply do not have the tools to see it. It would be the biological equivalent of dark matter, We know dark matter exists because we can see its gravitational effects, but we cannot see the matter itself. A shadow biosphere would be all around us, but biochemically invisible. Where could this shadow life have come from? It could be a true second genesis, a separate origin of life that happened right here on Earth. Maybe life started many times in many different ways, and the DNA-based life we know simply won out in the competition, pushing the other forms into obscure niches. Or, in the context of Panspermia, the shadow biosphere could be the alien life. Perhaps life from space landed on Earth, but it was so different from the native life that it could not compete effectively. It survived only in isolated pockets, or maybe our life is the alien life and the shadow biosphere is the true native life of Earth. How could we find it? We would have to start looking for life, as we don't know it. This means searching for strange metabolisms in unusual places. For example, we could look for environments that seem to have a lot of biological activity, but where we cannot find any familiar microbes. The Atacama Desert in Chile is one of the driest places on Earth. It is a good analog for Mars. Scientists have found strange pockets of life there that are not well understood. And, another approach would be to design tests to look for alternative biochemistries. We can search for proteins made of right-handed amino acids or genetic molecules that are not DNA. The discovery of a shadow biosphere would be one of the most profound discoveries in the history of science. It would prove that life is not a one-off fluke. It would show that biology is a universal phenomenon, something that can arise in different forms. It would mean that the universe is likely teeming with life, much of it very different from us. It would change our definition of life itself. The idea is highly speculative. There is currently no direct evidence for a shadow biosphere. But it is a powerful reminder of how much we still have to learn about our own planet. Before we can be sure that there is no life on Mars, we need to be absolutely sure we have found all the life on Earth. Number 4, our universe itself is a biological system. Most theories of panspermia treat life as a passenger. It is a fragile cargo that hitches a ride on a rock or a comet. The universe itself is seen as a vast, sterile, physical machine. Life is a lucky accident that happened in one of its corners. But what if this view is completely backward? What if the universe is not a physical system that happens to contain life, but a biological system that happens to contain stars and planets? This is the most radical and far-out version of the panspermia idea. It suggests that life is a fundamental property of the cosmos. This idea has been explored by a few thinkers on the far edge of science. The physicist Lee Smolin proposed a theory called cosmological natural selection. He suggested that universes are born from the black holes of other universes. Each time a new universe is created, its physical laws are slightly different. The universes that have laws that are good at producing black holes will have more offspring. He argues that the laws of our universe seem finely tuned to produce the stars that create black holes. But a side effect of having lots of stars is that you also get the conditions necessary for life. So life might be an inevitable byproduct of a universe that is good at reproducing itself. Another more biological take on this is the idea of a living universe. This theory proposes that the entire cosmos is analogous to a single living organism. The galaxies are like cells. The great voids between them are like the spaces between cells. The universe is born, it grows, and it reproduces through black holes, as in Smolin's theory. In this view, life as we know it is not an accident. It is a necessary part of the universe's metabolism or reproductive system. We are like mitochondria in a cell. We are a small but essential part of a much larger living entity. This might sound like philosophy or mysticism, not science, but there are some intriguing connections. The large-scale structure of the universe, with its filaments of galaxies and great voids, looks surprisingly similar to the structure of a neuron in the brain. The way information flows in complex systems, whether it is a brain, the internet, or the universe, seems to follow similar mathematical laws. In this context, panspermia takes on a new meaning. It is not about life accidentally spreading from planet to planet. It is the universe's way of propagating itself. The microbes on a meteorite are not just passengers. They are like seeds or spores of the cosmic organism. They are the mechanism by which the universe ensures that its genetic material, the potential for life and consciousness, is spread to every available niche. Fred Hoyle, again, touched on this idea. He was deeply troubled by the improbability of life arising by chance. He famously said the probability of life arising from non-life is comparable to the probability that a tornado sweeping through a junkyard might assemble a Boeing 747. He concluded that life must be the result of intelligence, but not necessarily an alien civilization in our universe. He proposed that the intelligence was the universe itself. He believed that the laws of physics were deliberately fine-tuned by a super intellect to allow for the existence of life. This is, of course, deep speculation. It is far beyond anything we can test with current science. It blurs the line between physics, biology, and philosophy, but it is the ultimate extension of the panspermia concept. It takes the idea that we are not from here and expands it to the idea that here is not what we think it is. It suggests that our quest to find the origin of life on Earth is too small a question. The real question might be, what is the role of life in the universe? If the universe is a living thing, then we are not just a cosmic accident. We are part of its machinery. We are a way for the cosmos to know itself. Number 3, the primordial soup was in an asteroid belt. The classic primordial soup theory, proposed in the 1920s by Alexander Oparin and J.B.S. Haldane, imagines life starting on Earth. In a shallow pool or ocean, simple inorganic molecules were transformed by energy from lightning or ultraviolet light into the complex organic molecules of life. This is the standard textbook story, but it has problems. The early Earth's atmosphere was probably not as reducing rich in molecules like methane and ammonia as the theory required, and a dilute ocean soup might not have been concentrated enough for the necessary reactions to occur. So some scientists have proposed moving the soup, not to another planet, but to the parent bodies of asteroids and comets in the early solar system. This is a variation of the pseudo-panspermia theory. It suggests that the crucial chemical steps that lead to life did not happen on Earth, but in space, inside the planetesimals that were forming about 4.6 billion years ago. These early asteroids would have been ideal chemical reactors. They contained all the necessary ingredients: water ice, simple organic compounds like formaldehyde and cyanide, and mineral catalysts like clays. The energy source would have been the decay of short-lived radioactive isotopes like aluminum-26. This decay would have heated the interiors of the asteroids, melting the ice and creating pockets of warm, liquid water. These pockets could have persisted for millions of years. Inside these watery asteroid interiors, you would have a perfect primordial soup. It would be a concentrated broth of chemicals protected from the harsh radiation of the young sun. The repeated cycles of freezing and thawing as the asteroid moved in its orbit could have helped to drive chemical reactions. The clay minerals would have provided surfaces for molecules to stick to, concentrating them and helping them to link together into long chains or polymers. This is a crucial step toward life. It is how you get from simple amino acids to complex proteins, or from simple nucleotides to RNA. Experiments in the lab have shown that these processes are plausible. Scientists have been able to create complex organic molecules, including the components of DNA and proteins, under conditions that simulate the interior of an early asteroid. In 2019, a team in Japan created ribose, the sugar in RNA, by simulating the reactions that could happen in an icy body. This theory neatly solves many of the problems with the Earth-based primordial soup. The ingredients are more concentrated. There is a long, stable period for the chemistry to happen. It also helps to explain the evidence we see in meteorites like Murchison. The complex organic molecules in that meteorite were not just picked up in space. They were likely formed inside its parent asteroid. The meteorite is a sample of one of these ancient chemical laboratories. After millions of years of cooking, these asteroids would have been filled with a rich slurry of prebiotic molecules. Some may have even taken the first steps toward life, forming self-replicating RNA molecules or simple cell membranes. Then, during the late heavy bombardment, these asteroids and comets would have rained down on the young Earth. They would have delivered not just a few simple ingredients, but a pre-made, complex, organic starter kit. Life on Earth did not have to start from scratch. It had a huge head start. It was like getting a cake mix delivered to your door instead of having to grow the wheat and sugarcane yourself. This theory is very appealing. It is a middle ground between a purely terrestrial origin of life and the more extreme versions of panspermia. It does not require living microbes to survive a long space journey. It only requires complex but non-living molecules to do so. It is strongly supported by the evidence from meteorites. It suggests that the potential for life is not unique to Earth. Any planet forming in a solar system will be seeded with these life-giving molecules. The origin of life may not be a planetary event, but a solar system-wide one. Number 2, the galactic scrapyard and reanimation. One of the biggest problems for panspermia is death. Even the toughest microbes will eventually succumb to radiation damage over a long enough journey. The DNA gets shattered, the cell machinery breaks down, a million-year trip through interstellar space is a death sentence. But what if death is not the end? What if a dead microbe can be brought back to life? This is the idea of necromanspermia, or panspermia by dead organisms. The theory was proposed by the astrobiologist Paul Wesson. He argued that even after a microbe is killed by radiation, its information is not lost. The basic chemical structures and the genetic sequence are still there, just broken. The dead microbe is like a smashed up blueprint. All the pieces are there, but they are not assembled correctly. According to this theory, the galaxy is filled with the dead remains of microbes. And comets and asteroids are like cosmic tombs carrying the shattered corpses of life from countless worlds. When one of these celestial hearses crashes onto a suitable planet, like the early Earth, it releases its cargo of dead microbes. On this new world, the conditions might be right for repair. The fragments of DNA and proteins from the dead aliens would be scattered in a primordial soup. Then a process similar to natural abiogenesis would take over, but instead of starting from simple chemicals, it would start with these highly complex, pre-made fragments. The local environment could act as a repair shop. The fragments of the alien DNA could serve as a template. These simple Earth-based molecules could line up against the alien template and form a new, complete strand of DNA. It would be a form of resurrection. The information from a long-dead alien organism would be used to jumpstart a new living organism. This solves the radiation problem. It does not matter if the microbes die on the journey. In fact, it is expected. The important thing is that their information survives. This makes interstellar panspermia much more plausible. The journey could take hundreds of millions of years. As long as the rock or comet eventually lands on a habitable world, the information it carries can be reanimated. This idea also has some interesting implications. It would mean that life on Earth is a hybrid. It would be a combination of alien information and terrestrial chemistry. It might also explain why life appeared so quickly on Earth. The process of reanimating a dead microbe would be much faster and more probable than creating a living cell from scratch. Is there any evidence for this? Not directly, but we know that DNA is a remarkably robust molecule. Scientists have been able to recover and sequence DNA from long-extinct animals like mammoths and Neanderthals. The process of DNA repair is also a fundamental part of all life. Our cells are constantly fixing damage to our DNA. The idea of using fragments of old DNA to build new strands is the basis of technologies like PCR, which is used in genetic labs every day. Necropanspermia is a speculative but clever solution to one of the biggest hurdles for the theory. It suggests that the galaxy is not just seeded with life, but with the ghosts of life. These ghosts are just waiting for the right conditions to be brought back from the dead. Life on Earth may not be descended from a living alien ancestor, but from its corpse. We might be the resurrected children of a billion-year-old cosmic graveyard. Number 1, we are the aliens, and we are seeding the galaxy. And panspermia theories usually look to the past. They try to explain how life got here, but what if panspermia is not just about the past? What if it is about the future? And what if we are the ones who will do the seeding? This is the idea of directed panspermia in reverse. We are not the recipients of a cosmic message, we are the senders. This idea starts with the Fermi paradox. The paradox, named after physicist Enrico Fermi, asks a simple question: if the galaxy is full of alien civilizations, where is everybody? The galaxy is old. There has been plenty of time for civilizations to arise and spread. Even with slow sublight speed travel, a civilization could colonize the entire galaxy in a few million years, a blink of an eye, in cosmic time. Yet we see no evidence of them. No giant alien structures, no radio signals, no probes. The sky is silent. There are many possible solutions to the Fermi paradox. Maybe we are the first intelligent civilization. Maybe interstellar travel is impossible. Maybe they are all hiding, or maybe they are all dead. The Great Filter theory suggests that there is some step in the development of life that is incredibly difficult. It could be the jump from non-life to life. It could be the jump from simple cells to complex cells. Or it could be the jump from an intelligent species to a star-faring one. Maybe civilizations inevitably destroy themselves with their own technology through nuclear war or climate change before they can get off their home planet. But what if we are the ones who make it through the filter? What if we are the first species in our galaxy to develop the technology for interstellar travel? If that is the case, then the future of life in the Milky Way could rest on our shoulders. It would be our responsibility or our opportunity to spread life to other worlds. We are already taking the first steps. We have sent robotic probes to every planet in our solar system. We have landed rovers on Mars. We are planning future missions to the moons of Jupiter and Saturn, like Europa and Enceladus, which may have liquid water oceans under their ice shells. These missions are supposed to be sterile. We go to great lengths to decontaminate our spacecraft so we do not accidentally bring Earth microbes to other worlds. We do not want to contaminate a potential alien biosphere before we even find it. But accidents happen, and in the long run, as humans begin to travel to these worlds, contamination will be inevitable. If we ever establish a colony on Mars, we will bring our entire microbiome with us. Mars will be seeded with Earth life. Now, think further into the future. Imagine a time when we can build interstellar probes. We could design a mission similar to the one Crick and Orgel imagined, but in reverse. We could build small robotic Genesis capsules. Each capsule could contain a diverse ecosystem of hardy microorganisms, perhaps extremophiles that can survive a wide range of conditions. We could build thousands of these probes. We could launch them towards nearby star systems that we have identified as having potentially habitable planets. The journey would take thousands of years. The probes would be designed to protect their microbial cargo from radiation. When a probe reaches its destination, it would enter orbit around the target planet. It would study the planet's atmosphere and surface to confirm it is a suitable home. If it is, the probe would release its cargo. The microbes would enter the atmosphere and seed the planet with life. Why would we do this? For many reasons. As a backup plan for life on Earth, as a way to ensure that the legacy of life continues no matter what happens to our own planet. Out of a sense of philosophical or even spiritual duty to spread the gift of life. It would be a project that would take thousands of years to complete. The people who launched the probes would never know if they succeeded. It would be a gift to the distant future. This is not just science fiction. Scientists and engineers have already sketched out the basic designs. for such missions. The technology to build small, long-duration interstellar probes propelled by laser sails is within the realm of possibility in the next century. The idea is called directed panspermia or galactic gardening. It recasts humanity not as a cosmic accident, but as a potential agent of cosmic evolution. All the other panspermia theories look to the stars and ask, where did we come from? This one looks to the stars and asks, where are we going? The ultimate far-out theory is not that aliens seeded us, but that we are the aliens who will one day seed the galaxy.